So today we're going to look at the structure and function of some of our biomolecules some more, and looking at how a change in the subunits of a polymer can lead to changes in the structure and function of the macromolecule. So starting off with our nucleic acids. Nucleic acids are a linear sequence of nucleotides, we can see here, that are defined by the 3' hydroxyl and 5' phosphates of the sugar of the nucleotide. So here's our sugar. Remember, it's got 5 carbons, so we can count our carbons. 1, 2, excuse me, that's the oxygen. 1, 2, 3, 4, 5 carbons, and figure out our directions. Within this, where our phosphates are, and during the DNA and RNA synthesis, nucleotides get added, going from the 5' end onward. And we'll get into DNA synthesis closer to genetics. That'll be a whole nother unit. This directionality influences the structure and the function, though, of the polymer. In the case of the nucleic acids, they're defined by their 5' and 3' ends, and that's going to determine which of the complementary nucleotides are added during DNA synthesis. This will also become important when we talk about transcription later on, which always occurs from the 5' to 3' end. These shapes are sort of, you can think of them, one's right side up, the other one's upside down. And so when we get into the enzymes involved in that process, the shape becomes important. With our nucleic acids, it's important to remember that DNA is structured, it's anti-parallel, so right side up, upside down, 2 different sides, in a double helix form. In this double helix, adenine nucleotides pair with thymine via 2 hydrogen bonds between them, so that's why in this image we've got 2 little dots. Cysteine pairs with guanine nucleotides by 3 hydrogen bonds, so it does take more energy to break apart a guanine and a cytosine compared to an adenine or a thymine. And just a reminder, here is our double helical shape of a DNA. Proteins are going to be linear chains of amino acids that are connected by the formation of covalent bonds between the carboxyl group of one and the amino group of the other. In this case, the hydroxyl group of the gets removed, as well as another hydrogen. We get water. This is an example of dehydration synthesis to form our bond. And so a protein starts as what we call a polypeptide chain, just a long chain of amino acids. However, for a protein to work, proteins have to be folded and they take on shapes. So proteins have a primary structure that's determined by the sequence order of their amino acids, a secondary structure that arises through local folding of the amino acid chain into either helices, so a single helix, or a sheet where it's just sort of folded up and down in a zigzag. Then there is a tertiary structure that is the overall 3-dimension of the shape. That's caused by the interactions of hydrogen bonding, and a little bit of van der Waals interactions are in there. And then lastly, most proteins, I'll say, have a quaternary structure, and that means that there are multiple polypeptide units involved. So for example, hemoglobin in your blood, there are 4 amino acid chains that form the entire hemoglobin protein when it's completed. That's the quaternary structure. So primary structure, this is a sequence of acids. Specific acids, they need to go in order to form the correct protein. If those acids get messed up, the case of hemoglobin, the disease sickle cell anemia, there is one amino acid that is different between a person who has sickle cell anemia, their hemoglobin is misshapen, versus a person who has regular hemoglobin. One amino acid messes the whole thing up. The secondary structure is, again, the alpha helix, which is a coiled shape, or the beta pleated sheet which is a more folded shape. And then we get our tertiary structure due to interactions between the R groups. Some R groups are going to be hydrophobic, so they don't want to be on the outside of the folded protein, they want to be on the inside. There's going to be van der Waals interactions. We're going to get hydrogen bonds between those that are polar and those that are nonpolar, as well as disulfide bridges. Some of our R groups have a sulfur atom on it. Sulfur will have an attraction to itself. So two separate R groups that both have sulfur, those sulfur atoms will want to be next to each other to form disulfide bridges. So two sulfurs will come together. And then the quaternary structure is when we have two or more of these polypeptide chains then group together, and they're going to group together and hold on together based on all of these different hydrogen bond and van der Waals interactions. We can also see this with carbohydrates, in which the carbohydrate polymer might be linear, just rows and rows and rows and rows and rows of a monomer, a monosaccharide again, or they can be branched. Again, this determines their structure and what they can do. We look at cellulose, it's very long and linear. That's what's in the cell walls of plants. If we look at starch, though, that's going to be more branching. That's a storage structure. All right. Thank you for joining me today.